all right, and the channels. Just a quick review. Pumps are an active form of transport across the plasma membrane. So that means they are going to require energy, and it also means that it's going to move substances against their concentration gradient. All right. Now the important thing is we're going to learn about this concentration gradient, all right, in this chapter, but it's the pumps that actually will maintain. They make the concentration gradient. All right. Remember the banana floating in the ocean? You folks didn't get to see that. You probably heard it when I was talking. Okay. So here's the banana. All right. Rich in potassium, and then it's floating in the ocean. That's me again. I apologize, y'all. I'm bad. Leave my phone. All right. And then it's floating in the ocean, which is high in sodium and chloride. Salt water. Okay? So basically what this represents, the banana represents the cell. Has a lot of potassium inside. Okay? And then this bigger circle is the ocean. And that represents your ECF, extracellular fluid, or also known as the interstitial fluid. All right? And then inside the cell is known as the ICF, intracellular fluid. Okay? So outside of your cell, you've got lots of sodium, lots of chloride. All right? You don't have much inside the cell. But inside the cell, you got a lot of potassium. You just don't have a lot outside. So this sodium, the pumps here, there's two pumps, okay, when we're dealing with a neuron. You've got a calcium pump, and that's going to be more towards the synaptic knobs at the end of the axon or the neuron. And that's going to be pumping calcium out of the cell. So there'll be more calcium outside the cell. Okay? Our sodium potassium ion pump, which we saw in chapter 4, is going to pump potassium into the cell, and it's going to pump sodium out of the cell against the concentration gradient. Okay? So that's, and as it's doing that, it's maintaining that concentration gradient. It's keeping more sodium outside the cell, all right, or it's trying to, and at the same time, it's keeping more potassium inside the cell. Because if we were to turn the pumps on, off and open up leak channels, because they're, well, we don't have to open them up, they're always open, all right? Then sodium is going to want to go in what direction? If I put a hole here in the cell, what's sodium going to want to do? If it's at high concentration outside the cell, low concentration inside the cell, what does it want to do? Yeah, it's going to want to diffuse, all right? It wants to go in. Sodium will go into the cell. It's going to diffuse down its concentration gradient, right? If we have a lot of potassium, all right, inside the cell, and I put a potassium leak channel in, where's potassium going to go? Out. Yeah, right. It's going to leave. Right? Because there's more potassium inside, less outside, it's going to diffuse down its concentration gradient. Which brings me to this guy. Channels. Okay? Channels operate passively. They're a passive process. Okay? So that means they don't require energy, all right? But they do move substances, all right, down the concentration gradient. From an area of high concentration to an area of low concentration. All right? So we're going to go down the concentration gradients. But these channels are specific to one type of ion. So if it's a potassium leak channel, only potassium can go through it. If it's a sodium leak channel, only sodium can go through it. Okay? These channels are ion specific. All right? Calcium channels, only calcium can go through. Are we good on that? Channels are specific. Okay, so the first type are the leak channels. These guys, remember this is the one I told you is 7-Eleven. It's open 24/7, open all the time. All right, so they're going to let sodium and potassium diffuse across the plasma membrane down their concentration gradient. That means potassium will leave the cell, sodium will enter the cell. Okay, these next two channels here, the first word in their title tells you what we need to open that channel. These channels are normally closed. Okay? So we need a stimulus. Okay? So, for example, the chemically gated channel, we need some sort of chemical to open this channel up. And in this case, it's going to be a neurotransmitter. All right? That's the only chemical that we're dealing with right now. So a neurotransmitter will bind to this channel, and it opens up the gate, okay? And a specific ion will move down its concentration gradient. So chemically gated sodium channels tell me that I need a neurotransmitter to open up that channel, and once it opens up, sodium moves across the plasma membrane. Okay? Chemically gated potassium channels, same thing. I need a chemical, a neurotransmitter, that will stimulate and open up that channel, and then potassium will move down its concentration gradient. Chemically gated chloride channels. All right? The last one here, again, another gated channel, closed. Okay? But in this situation, the stimulus we need is a change in the electrical charge. And we're going to talk about what that means. Okay? The change in the electrical charge of the plasma membrane. Because believe it or not, your plasma membrane, or the neuron cell, has an electrical charge to it. Okay? Normally inside the cell, it's negative, and outside the cell, it's positive. Okay? When I say normally, when it's at rest. When the neuron's not doing anything and it's just relaxing, nothing's happening, all right? It's positive outside, negative inside. We'll talk why that is, okay, when we get to the membrane potential. So in this situation, we're going to need a change in the charge across the plasma membrane to open up these channels. So if I'm talking about a voltage-gated sodium channel, all right, when we get a change in the electrical charge across the plasma membrane, all right, this channel will open up and sodium will diffuse down its concentration gradient. And that's what we talked about last on Tuesday. All right, the voltage-gated sodium channels. This is the one that's got two gates. It's very unique. Okay, has the activation gate, which is that swing arm gate, and then the inactivation gate, which is that bulging here. Okay, so that's what we're seeing here in this picture. Here's the activation gate, that's swing arm. Then in the background here, you can see these two bulges. All right, but they're not very big right now. So it's inactivated. The inactivation gate is open. The activation gate is swung closed. Okay, so nothing can move across. So in the resting state of our voltage-gated sodium channel, all right, the activation gate is closed, the inactivation gate is open, and you can see the outside of the cell is positive, the inside of the cell is negative. And so these purple balls are sodium molecules. All right, they want to come in because guess what? Look, we see 5 outside and 1 inside. There's more out than there are in. So it wants to enter in. Okay? All we have to do is give it a means to do that. Well, guess what? Now look, in the second phase here, all right, in the activation phase, all right, look what happened to the charge. We flipped it. Now the outside of the cell is negative, the inside of the cell is positive. We've changed the charge of the cell. We've affected it. Okay? And by doing that, we've opened up the activation gate. It swings open. The inactivation gate is still open, so sodium diffuses down its concentration gradient. And it moves into the cell. Okay? Then what will happen is, all right, we'll see a moment where the inactivation gate closes. So those little swellings just get bigger and they pinch off. So now sodium can no longer enter into the cell. The activation gate is still open. This is very brief. This phase is very, very brief. All right? So nothing moves across. And then what will happen is we return back to phase 1, the resting phase or state. Yep, it'll reset itself. But it needs a stimulus, and it needs that change in the voltage across the plasma membrane. And we'll talk about exactly all the specifics. This is kind of a general take on it. We'll get to, I'll walk you through all the steps. Okay? But you need to know, all right, that this type of, it's unique. Okay? The voltage-gated potassium channel is not like this. It doesn't have 2 gates. Okay? Only the voltage-gated sodium channel does. So it goes through all these states here, the resting, the activation, and then the inactivation. Okay? Yeah, Laney, that's a great question. I'm going to tell you about that. But what will cause that change is an action potential. When that electrical signal starts to move down the axon, we're going to change the voltage, and I'll get into the specifics of that. But to answer that question, yes, an action potential will cause that change in voltage. Okay, so let's jump in. That was just a review. So let's jump into today's information, okay? All right, so when we talk about modality-gated channels, all right, these types of channels, again, usually closed, all right, what we need for them to open up is a stimulus. And so depending on whatever that stimulus is, all right, it will affect a gate that is able to monitor that stimulus. Meaning, okay, if it's a chemically gated channel, then we need a chemical to open it up. A voltage change won't open that channel up. Okay? It has to be specific for that type. That's what we refer to, that's what we call a modality gated channel. That modality, whatever that stimulus is going to be, has to be specific for that channel. Just like a change in voltage won't open up a chemically gated channel. Okay? So the stimulus needs to match the modality for that channel. Okay? So we're going to see this in abundance in our sensory neurons. Makes sense. Because sensory neurons convey that sensory input from the internal and external environment. Okay? So we'll see this, all right, when we talk about the skin. We'll get into some of the specifics here, like for touch. Okay? So that mechanical pressure, all right, on the receptor, when you touch something, all right, that will be the stimulus. That mechanical pressure will then stimulate, all right, chemicals. If it's a Merkel receptor, it will release chemicals that will stimulate the sensory neuron in the vicinity, and it'll open up our chemically gated channels to then create an action potential, which hopefully, if we have time, we'll talk about that today. Okay, so let's talk about where we're going to find all these pumps and channels that we just learned about. All right, if you have your book in front of you, this is an awesome diagram here. Okay, it's figure 1212. It's cut off here, but this is, all right, this is the bottom portion of it. This is the top portion of it. Okay, and I'm going to go through, but you can see, all right, when we're talking about, all right, well, let me zoom in just a little bit. When we're talking about, all right, the entire length of the neuron here, you're going to find sodium, potassium ion pumps, and we're going to have our leak channels throughout the whole neuron. That includes the cell body, the dendrites, the axon, all right, all the way down here, okay, to our teledendra and the synaptic knobs. Okay, so those three types of channels, okay, or not channels, but those three types of transport proteins: sodium leak channel, potassium leak channel, and the sodium potassium ion pump. That's going to be located throughout the whole entire axon. Now, or neuron. You want to make sure that you're familiar with where these pumps are, okay? Because it'll make learning what occurs at these specific segments much more easier for you, okay? So if I go back up to here, okay? So across the entire neuron itself, all right, these pumps, well, this pump and these two channels are responsible for this right here. Maintaining the resting membrane potential. What the heck is that? Well, let me tell you, I'm going to give it a number, all right, in the beginning. It's negative, you need to know this, you need to memorize this, negative 70 millivolts. That is what we call the RMP, the resting membrane potential. What is that? That is the electrical charge across the plasma membrane when the neuron is doing nothing, when it's sitting there. When you're sleeping, all right, your motor nerves should not be causing your skeletal muscles to move, all right? So the resting membrane potential in those neurons, for those nerves, should be negative 70 millivolts. So if we were to go back up to this previous slide here, where we saw all these gates here, remember how I told you the inside of the cell is negative? It's actually negative 70 in a resting membrane, in a resting neuron. I'm going to give it a number now. Okay? So in the resting state, for this channel, the neuron's not doing anything. All right? The inside of the cell is negative 70 millivolts. And we refer to that as the resting membrane potential. We'll get into more of the specifics. But you need to know, I've seen test questions asked about this. What contributes to the resting membrane potential? Sodium-potassium ion pumps and the sodium-potassium leak channels. Number 2, important fact off of this slide here. There are way more potassium leak channels than sodium leak channels. And there's a big significance for that, right, which we'll touch on. Okay? So basically, sodium can diffuse that much easier across the plasma membrane than, potassium can diffuse that much easier across the plasma membrane than sodium because there's more leak channels for it. Okay? I'll explain the purpose for that. Okay, so we talked about the actual anatomical parts of a neuron. All right, the cell body, the axon hillock, the axon, the dendrites, all right, and the telodendron with the synaptic knobs. Now, we're going to talk about and describe those same anatomical structures, but we're going to talk about their functional descriptions, okay? So when we talk about the functional descriptions here, all right, we're talking about their physiology, all right? When we talk about the receptive segment, we're talking about the dendrites and the cell body. Okay? That makes sense. We call it the receptive segment because that part of the neuron is going to receive input. Okay? Information is going to come into that region there. So, on the receptive segment, we have chemically gated ion channels. Chemically gated. In addition to, I'm going to come right back to that slide. In addition to these channels and pumps. Because remember, these are the entire neuron. These are everywhere. Okay, sodium-potassium ion pump and our sodium-potassium leak channels. They're everywhere. Okay, but now we're going to the receptors. We're going to go to the receptive segment. Well, here's the receptive segment right here. Okay, the receptive segment. Okay, and that is going to have just chemically gated channels in addition to those 3 other structures that I just told you about. So an example would be a chemically gated potassium channel, right? Chemically gated chloride channel, chemically gated sodium channel. Okay? So remember, the first word is the modality. And then if you see a letter, that's what's going through the channel. Okay? So the next part, remember this, the initial segment, right? Remember the axon hillock? That's where the action potential starts. All right, that's this section right here. Okay, that little triangular region there. All right, so the type of channels that we see there are voltage-gated sodium and voltage-gated potassium channels, in addition to the sodium-potassium ion pump and the leak channels. Okay. This is important because what is significant about the initial segment or the axon hillock? What's really significant about it? That's where we start the nerve signal, the action potential. That's where it's made. That's where it starts. Okay? So we're involving these voltage-gated channels. Okay, so the next segment of our neuron is the conductive segment. All right, that makes sense. Like an electrical signal travels through a wire, the action potential, the nerve signal is going to travel through the axon and its branches. Now remember, some axons have collateral, so they'll pop off from the side. They'll just branch off. Okay? So the conductive segment is also going to have voltage-gated sodium channels and voltage-gated potassium channels, in addition to the sodium-potassium ion pump and the leak channels. Okay, so I'll show you a quick picture. Here is the conductive segment. Here's the axon. Okay. All right, then the last part is going to be the transmissive segment. We call it the transmissive segment because it's going to transmit our nerve signal onto something else. Maybe it's another neuron. Maybe it's an effector organ, like a muscle or a gland. Okay? So we get down to the far end, that's going to be where the synaptic knobs are, and we got something different down there. You heard me talking about the calcium ion pump, so now we know. All right, it's way down at the far end. And we have our voltage-gated calcium channels down there. Okay? That's this picture right here. So just all the way down here where the synaptic knobs are. That's the transmissive segment. So it's going to transmit our nerve signal, our action potential, onto something else. Okay? I would go into your book and just memorize that picture. You know what I mean. You know, just say, know what is where. And as we go through this chapter, I'm telling you, you'll I've talked about active and passive learning, but you will almost passively learn where all these channels are, just because of how much we're going to talk about. Okay? All right, so a quick question here. What is the difference between a chemically gated channel and a voltage-gated channel in terms of how they function? All right, chemically gated neurotransmitters are used, all right, to open up the channel. In voltage-gated, we need a change in that electrical charge. Remember, our resting membrane potential is negative 70 millivolts, okay? So we need to change that value. Maybe we need to make it negative 40. Okay, negative 20, negative 10, whatever. Okay, we need to change that value. Okay, that's supposed to be a 7, by the way. Okay, this next slide took me a long time to make. Okay, so I really want you, and I'm serious when I say this, that this next slide is that up there on the board, the banana floating in the ocean. Seriously, check it out. All right, it's nothing really exciting, but it explains the distribution of our ions, all right, calcium, potassium, sodium, chloride, where they are. Are they inside the cell or are they outside the cell? Okay? And it's because of this distribution, all right, that distribution is essential. It's vitally important for the proper functioning of the neuron. Okay? So, the first part here talks about the inside of the cell. All right, you heard me talk about potassium ad nauseum. Yeah, potassium. I know there's a lot of potassium inside the cell. Okay, banana, get it. But I haven't talked about, all right, our phosphate ions and our proteins. Now, you notice how I told you in the resting membrane potential, all right, why it's more negative on the inside of the cell versus the outside of the cell, in case you were wondering. All right, if not, I'll explain it to you. All right, it's because of these two guys right here, the negatively charged phosphate ions and the negatively charged proteins. That's what makes the inside of the cell negative. All right, and then you're probably thinking, well, Dr. Kaz, potassium's up there. All right, potassium is a cation ion. It's positively charged. You're absolutely right thinking that. But there's way more negatively charged proteins and negatively charged phosphate ions that they cancel out all the positivity of the potassium. Okay? And that's what makes it the inside of the cell more negative in a resting neuron. Okay? Here's the thing. These two structures cannot leave the inside of the cell. They're trapped there forever. For right now, okay? They can't leave. They're stuck there. Guess what? The only thing that can leave is this guy. The negatively charged proteins are way too big. There's no channels for them. The only way they're leaving the cell is if the cell explodes, if it lyses, if it dies. So they're stuck there. Same thing with the phosphate ions. They're not leaving because guess what? Your cells love phosphate. Why? Why do cells love phosphate? It's important. Energy function, ATP, okay? ATP adenosine triphosphate. Right? It wants to keep phosphate on hand because it's constantly recycling phosphate to make ATP. So it's not going to let it leave. Okay? So these two substances don't leave the cell. So they keep their negative charge there. The only thing that can come or go is the potassium. Okay? All right. The outside of the cell is way easier. Okay? The interstitial fluid, sodium and chloride. Now, granted, yes, I know what you're thinking, but Dr. Cobb, all right, chloride's negative. Yep, you're right. It is. And sodium is positive, but there's way more sodium outside the cell than chloride. So one sodium ion and one chloride ion will cancel each other out, okay, with their charge. But there's like 140 sodium outside the cell and like 20 chloride. So see what I'm saying? That's how we get that difference in charge. But you need to know chemically, okay, what is inside the cell, all right, potassium, and what's outside the cell, sodium and chloride. You feel pretty good about that? That you know what's inside the cell and what's outside the cell? Banana floating in the ocean. Okay, banana floating in the ocean. Okay, so now we're going to talk about a concept here in which we need to understand so we could figure out how the heck we get this resting membrane potential. How do we get this negative 70 value? Okay, let's talk about that. First, all right, we're going to talk about what we call the electrical chemical gradient. All right, we understand about concentration gradient, right? How there's more of one thing in one area and less of it in another area. Well, things will move down that concentration gradient. Now we're going to talk about a different type of gradient. We call it the electrochemical gradient. So it has two parts. It's going to involve electricity, so charges, and it's going to involve chemicals, okay, stuff, substances. So we're going to break it down, and we're going to talk about the electrical part first, okay? So we're going to talk about the electrical gradient, and here it is. In all its simplicity, it's the difference in the electrical charge between two areas. Okay, so what two areas are we talking about? We're going to talk about the cell, the inside of the cell and the outside of the cell. So the outside of the cell is positive, the inside of the cell is negative. That's what we're talking about. All right, when we talk about the electrical gradient, it's the difference in charge between the two areas. Okay, so the total charge, all right, outside the cell will be positive. All right, and the total charge inside the cell will be negative. Okay, so this part right here, you should know. Okay, positive outside, negative inside. All right, and if you're not sure how that is, refer to the previous slide. Okay. The distribution of these substances in the cell and outside the cell will help to generate, all right, this electrical gradient here. Okay? So this difference in charge inside the cell versus outside the cell, all right, we refer to that as the membrane potential. Okay? And we can alter that membrane potential. We can make it bigger or we can make it smaller. And I'll tell you how we do that here in a few. Okay, so that's the first part of the electrochemical gradient. The second part of it is the chemical, which is easier because guess what? You already just learned what the chemical gradient is on that first slide that I was showing you, all right, about the distribution of substances. All right, when we talk about the chemical concentration gradient, again, we're talking about the distribution, all right, between two areas, all right, but specifically the unequal distribution. Okay, how there's more in one area and less in another area. All right, banana floating in the ocean. We have way more potassium inside the cell than outside the cell. And we have way more sodium outside the cell than inside the cell. Okay, so because of that, we've created a concentration gradient, yes, but we also have a chemical gradient. We have that unequal distribution of those chemicals. We have more sodium outside the cell. We have more potassium inside the cell. Okay? So that is our chemical gradient. telling you, when you're taking your test, if you remember banana floating in the ocean, you're going to be good. You're going to get, all right, I just got to cut that out. I keep saying that. All right, so now, all right, now let's figure out, all right, what is going to happen. Because all this I'm building up to getting you to understand about what an action potential is. But what we need to have you understand is what an electrical current is and how it involves neurons. Okay? So the only thing up to this point that we really know that has an electrical charge, all right, is what we learned in chapter 2. All right? And the only things that have an electrical charge are going to be ions. Okay? So what are ions? They're charged particles. Okay? Cation is a positively charged ion, and an anion is a negatively charged ion. All right? So whatever that distribution is across the plasma membrane is going to affect that charge. So if you think about it, there's way more sodium and its positive charge outside the cell. Okay? And there's very little sodium inside the cell. All right? Even though there's potassium inside the cell, there's not nearly as much potassium inside the cell as there is sodium outside the cell. Remember what I told you, those negatively charged proteins and those negatively charged phosphate ions are pretty much going to dominate the negative charge contribution inside the cell. Okay? So this difference that we see across the plasma membrane, all right, we mentioned it on the previous, or 2 slides ago, is the membrane potential. Okay? So if we did not have, all right, and if you've taken a physics course, you're familiar with how voltage and amperage and how resistance works. Okay? So the fact of the matter is, when all the gates are closed, our voltage gates, our chemical gates, all right, it's more difficult for sodium and potassium to move across the cell. Okay? Even though they're going through the leak channels. Okay? But at the same time, as they're going across the leak channels, the sodium-potassium ion pump keeps pumping it out. All right? That whole process helps to develop this membrane potential. Okay? Remember, I told you, all right, when we have those leak channels and that sodium-potassium ion pump, it's going to maintain that concentration gradient there. Okay, keeps more sodium outside the cell. Right, sodium's positive. Keeps more potassium inside the cell, but that potassium gets canceled out, all right, by those negative charges inside. So the plasma membrane is going to act as a resistor or create resistance because one, remember, the plasma membrane is a semi-permeable. Well, I shouldn't say that. It's a limiting barrier. It only lets certain things cross the plasma membrane. Right? It has to be really small or nonpolar. Well, guess what? Ions aren't going to fit into that category. So they need to get across the plasma membrane through a channel. And if those channels are closed, it's going to create more resistance. So if we want to decrease the resistance, we either put more leak channels into the plasma membrane, or we open up some of our chemically gated or voltage gated ion channels. Okay? So when we have channels open, we decrease the resistance. When we have channels closed, we increase the resistance. Okay? And I'm not trying to get into physics here, all right, because this is a biology class and not a physics class. But, all right, when we get movement of ions, all right, movement of charged particles, we are actually going to generate energy. It's called kinetic energy. All right? If you ever took a physics class, even in middle school, we have a hill here, and at the top of the hill we have a boulder. At the top of the hill, that boulder's not moving, all right? It's what we consider potential energy. Because if I push the boulder, it will start to roll down the hill. When it's actually in motion and rolling down the hill, that's kinetic energy. Okay? So when our ions actually are moving across the plasma membrane, they're going to generate energy or current. Okay? Current is recorded in amperage, okay, for the value. Don't worry about that. All right? So as the ions move across that plasma membrane through the open channels, they will generate a current, a charge. All right? We're going to talk about that here in a second. All right. What is the electrical gradient? All right, the electrical gradient is going to be the electrical charge difference between two areas. In this case, our two areas are inside the cell and outside the cell. Chemical gradient, a little bit easier. Unequal distribution of those substances between the two areas. Same thing. All right? That chemical gradient is going to be attributed to the fact that we have more sodium outside the cell than inside the cell, or more potassium inside the cell than outside the cell. All right. If you're still not 100% on this, not to worry. We're going to break it down even easier now. So what we're going to do is we're going to talk about what is going on in a resting neuron. All right. So you're going to see, you already understand some of what I've told you here already. Okay. When a neuron is at rest, you're going to see that their ion concentration is going to be unequal in and outside the cell. All right. So the fact that we have more sodium and chloride, well, we even throw calcium in there. All right. That will be a higher concentration, all right, in the interstitial or extracellular fluid, all right, outside the cell than in the cytosol, which is inside the cell. And we'll see more potassium inside the cell or in the cytosol versus the interstitial fluid. All right, do you feel comfortable knowing where the distribution of chemicals are in general? Yes, no? Okay, so more sodium outside the cell, more chloride outside the cell, all right, and more, in this case, we're not going to talk about calcium too much until we get to the end of the neuron at the synaptic knobs there. All right? All right, so when the gates are closed, which they should be at a neuron at rest, except the only gates that are open, what are the only gates that are open when a neuron's at rest? They're open all the time, 24/7. Leak channels. Yeah, leak channels are always open. They don't have a gate on them. Okay? So you can't, even when the action potential is generated, when all this stuff that I'm going to talk about here soon is happening, the leak channels are always open. They're always letting sodium and potassium diffuse across the plasma membrane. But remember, okay, the more channels that you have open, okay, the more chemical or ions can flow across the plasma membrane. It's when they're closed, all right, that they're going to meet resistance. All right. So here's the situation. This gives us more positive ions outside the neuron than inside of it. So that means, here's our plasma membrane, that outside is going to be positive, inside is going to be negative. All right? Because there's more positive ions outside, all right? And there's fewer positive ions inside. So that gives us that difference in the electrical charge across the plasma membrane, all right? And that's what we refer to as that membrane potential. All right? Important fact here, I'm going to reiterate it, all right? The cytosol is relatively negative because phosphate and proteins, all right, cannot cross over the plasma membrane. There's no channels, there's no gates for them. So they're stuck inside, and they're negatively charged. So they're going to defer their negative charge to the inside of the cell. Okay? So when the neuron is at rest, what we're going to do is we take this instrument here called the voltmeter, all right, and it's got two leads. We stick one lead inside the cell and the other one outside the cell. And this measures, all right, the voltage, all right, or the electrical gradient across the plasma membrane. And that's the value that we get, negative 70 millivolts. We didn't make it up. We actually used an instrument to figure that out. Okay? So this gets asked all the time on a test. Resting membrane potential. Okay? Can we all at least say that you feel good that you know that number? Okay, all right. So we know the number. Good. All right, so I'll make it simple for you. This is how we figure out that number. All right, see this bullet point here. There's more positive ions on the outside of the neuron than inside. Okay, so that makes the outside positive, the inside negative. All right, that difference in charge between the outside of the cell and the inside of the cell is going to be this value here. Okay? So what makes it more positive outside? These ions. Okay? That's what makes it more positive outside. What makes it more negative inside? Phosphate and proteins. Okay? That's what's going to contribute mainly to the negative charge on the inside of the cell. Okay. All right, so continuing on here, now we know what this is, this resting membrane potential. All right, when the neuron is not doing anything. So this next statement here, make sure you star this. Potassium diffusion is the most important factor in setting the resting membrane potential. All right. At the beginning of class, do you remember when we were talking about leak channels? And I said that there's way more potassium leak channels than sodium leak channels. And I said to you, that's important. And we're going to learn why later on in the class. Well, guess what? It's later on in the class, and now we're going to learn why. Okay? So, if we open up, oh, well, I don't even have to use that example. We have leak channels in our neuron. Okay? It gives the opportunity for potassium to move across the plasma membrane. Knowing what we know about the concentration gradient, all right, like I showed up here on the board, and it's still up there, where does potassium want to go? Through the leak channel. It has an opportunity to go through the leak channel. Where's it going to go? Is it going to come into the cell, or is it going to leave the cell? It's going to leave why? Diffusion. Diffusion. All right? It's diffusing across the plasma membrane. It's going to go from that area of high concentration to low concentration. We call that moving down its concentration gradient. Okay? Now here's the thing. Look at the charge of potassium. What charge is that? Positive, right? Okay. What do we know about opposite charges? Opposites attract, right? Okay. So, as potassium starts to leave through the leak channel, okay, in the beginning, while its concentration gradient is really steep, all right, well, there's a huge, remember on our little chart there, okay, the concentration gradient is really steep. Okay, so that means the steeper the concentration gradient, the faster it'll diffuse. Okay, if you have if the incline for the concentration gradient is less steep, then it's going to diffuse slower. I tell folks, just think of it like this. It's like rolling a ball down a hill. Won't it roll faster down a steeper hill than it will a more shallow hill? All right? Okay? So that's what's going to happen with potassium in this case. It's going to diffuse out real fast in the beginning. It's going to roll out of there. It's going to try to get out of there. But guess what? The inside of the cell is negative. Why? What makes the inside of the cell negative? Exactly. The negatively charged phosphate and the negatively charged proteins, they're negatively charged. Potassium is positively charged. All right? Now, the concentration gradient in the beginning is so big, they just rush out of there. But as it starts to get less, it starts to slow down. Well, the negative charge of those proteins and phosphate start to pull on the potassium. Remember, opposites attract. So it's trying to get out of there, but yet the positive force, the attractive force of the negative ions will start to pull it back in. Okay? So that's what we're going to see here. We say that it's limited because of the electrical gradient. Well, what's that? That's the pull of the negativity, all right, of the inside of the cell from the phosphates and from the proteins. Okay? And so what we'll see is as potassium starts to, in the beginning, all right, it will pour out fast, but because the concentration gradient is so steep, okay? But then as that steepness declines, okay, to this, then the negative attraction from the proteins and the phosphates overcomes the concentration gradient, and it becomes harder for potassium to diffuse out. So that's what we're talking about. The electrical gradient, opposites attract, all right? That becomes greater, all right, as more potassium leaves, and it starts to slow down its diffusion. Okay? And think about this too. As more potassium leaves the inside of the cell, remember potassium's positive. Our negative charged items can't leave. So as more potassium leaves the inside of the cell, we start to see the inside of the cell become more negative. That means its negative attraction for potassium becomes stronger. So it makes it even harder for potassium to leave the cell as time goes on. Okay? So it slows down its diffusion. Because remember, we want equilibrium. We want, all right, we go from this steepness grade like that, and then it starts to decrease like that for the concentration gradient, and then eventually it just becomes balanced, equal. 55 molecules on the inside, 5 molecules on the outside. Okay? But as it becomes, or as we approach equilibrium, the negative attractive forces from the proteins and the phosphates becomes greater and greater, so that attractiveness becomes more and more, so it slows the diffusion of potassium out of the cell, okay, as we get closer to equilibrium. Just to let you know, this PI represents inorganic phosphate, in case you're wondering. Okay? So, here's the thing. If, and this is a fact, and this is something that you definitely want to know, okay, if we only had potassium leak channels, our resting membrane potential would be a lot different. It wouldn't be negative 70 millivolts. The reason why it's negative 70 millivolts is on the next slide. We're going to talk about that. The sodium leak channels. All right, if it was just potassium leak channels, our resting membrane potential would be negative 90. Okay? Negative 90. If we did not have any sodium leak channels, because guess what? We know what's going to happen with sodium. Leak channels in our cell, it's up here on the board. All right, sodium wants to enter into the cell, right? It wants to go down its concentration gradient here. That's what we're going to see, all right? Sodium is going to leak into the cell, all right? It's going to go down its concentration gradient. Here's the interesting fact, though, okay? Sodium is positively charged. What's the charge of the inside of the cell? Negative. Remember, opposites attract. So not only is sodium, all right, going to go into the cell through its leak channel to move down its concentration gradient, it's getting pulled in. It's getting help. Okay? So that pull from the electrical chemical gradient that is from the proteins, I'll call them the pro, and the inorganic phosphates, all right, they're negative. Okay? They're going to pull sodium in. Okay? But here's the sad reality. If we had equal amounts of sodium and potassium leak channels, our resting membrane potential wouldn't be negative 70. It would be less negative. Okay? But because we only have a few sodium leak channels, all right, the value is at negative 70. Okay? If there were equal amounts, it'd be less negative because think about it. Sodium's getting pulled in, it's getting helped. Right? Potassium, when it's trying to leave the inside of the cell, you know, in the beginning it's flying out because the concentration gradient is so great. There's so much of it, it's pouring out. Then when this concentration gradient becomes less and less and less, there's not as much incentive, all right, for potassium to diffuse out. And then it makes it even harder because the negative attractive forces of the proteins and the inorganic phosphate are trying to pull it back in. All right? Okay. So staying here with our neuron at rest, let's talk about our last structure that helps to influence the resting membrane potential. Because remember, sodium-potassium ion pumps and leak channels maintain, all right, the concentration gradient, which influences our resting membrane potential here. All right, so you want numbers? For every ATP that this pump uses, it's going to pump 3 sodium ions out of the cell, while at the same time, it's going to pump 2 potassium ions into the cell. All right? Sodium potassium ion pumps play such a small role, they only contribute a -3 millivolts to the overall -70 millivolts. So what you should gain from that is the sodium potassium ion pump plays such a small role in determining the resting membrane potential. It plays a large role in maintaining the concentration gradient because it's pumping sodium and potassium in and out of the cell, but it only, for its role, only contributes -3 millivolts. The majority comes from potassium leak channels. All right, and then the next most influential is the sodium leak channels. And then what's really hardly of consequence are the sodium-potassium ion pump. But what is important is that it maintains the concentration gradient, all right, for sodium and potassium. And what's even crazier is that it takes up 66% of a neuron's energy usage. Just operating this pump all the time. As you're sitting there, these pumps are operating. Right? And it's gobbling up ATP. It's part of what we call the basal metabolic rate. Just to maintain this concentration gradient, right? To maintain a resting membrane potential of -70. That pump does a lot of work, but it just doesn't really influence the resting membrane potential all that much. It does influence the concentration gradient. You need to know the difference between that. Okay, what's the concentration gradient? More sodium outside the cell than inside the cell. More potassium inside the cell than outside the cell. That's the concentration gradient that we're talking about. All right, it plays a huge role in that. The leak channels affect the resting membrane potential. That charge, okay? So here you can see pretty much what I've been talking about, okay? You've got your sodium-potassium ion pump. It's pumping sodium out of the cell, pumping potassium into the cell, and then we have our leak channels, letting potassium leak out and sodium to leak in. Always, always, always going down their concentration gradients. And meanwhile, all right, we've got our inorganic phosphate and our negatively charged proteins, all the while, which are going to exert their negative charge on pulling potassium back into the cell as its concentration gradient starts to flatten, and at the same time, it will pull sodium into the cell as it diffuses across the plasma membrane. Okay? Okay, so this is now. Keep in mind, let's not forget about the other players here, okay? In a resting neuron, I don't have it up here. So at our resting membrane potential, which is that -70 millivolts here, what are the other channels doing? Okay, well, all right, our voltage-gated sodium channel is closed. Our voltage-gated potassium channel is closed. And our voltage-gated chloride or chemically gated chloride channel is closed. All right? So that's what's going on. All those channels are closed. They're not doing anything. Now we're going to talk about what happens when they open up. Okay? So the resting membrane potential is primarily established by what two structures embedded in the plasma membrane? Resting membrane potential, RMP. Okay? Sodium ion and potassium ion lead channels. Which is more influential on the resting membrane potential? Potassium, right? Potassium, 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 which is K+. Okay? They play a huge role. All right. I know this is a lot. This is a tough chapter. That's why I go through it really slow. It doesn't seem like I'm moving slow. Believe you me. I would love to have 5 classes to teach this, but I'm only given 3. So I'm gonna teach you some definitions now. Okay, let's just go some straight memorization here. All right, real easy. These definitions are crucially important because you need them, because when we do chapter 10, I'm going to be using a lot of these terms. Okay, so the first term that we're going to talk about here, well, the 2 terms we're going to discuss are depolarization and hyperpolarization. Right, it involves the opening of our chemically and voltage-gated channels here. And when we open those channels, things are going to move in and out of the plasma membrane. Okay? What does that mean? We're going to change our resting membrane potential. What does that mean? We're going to somehow change this number. Okay? We're going to change that negative 70 millivolts somehow. Okay? So let's talk about the first way we're going to change it. Depolarization. Okay? Depolarization. Look at the definition. All right, well, first of all, this only occurs at the plasma membrane, but this is what happens when we make the inside of the cell more positive than the resting membrane potential. Or I could say, this is what happens when we make the inside of the cell less negative. Okay? So, is negative 80 millivolts more positive or less positive? Or let me see, that's an awful way for me to phrase that. Is negative 80 millivolts more negative than negative 70 or more positive than negative 70? More negative, yeah, okay? Okay, good. So is negative 60 less negative than 70 millivolts, negative 70 millivolts, or less positive, or more positive than negative 60, than negative 70? Right, it's less negative. All right, good. Okay, I just want to make sure that we're all on the same page here. So, when we make the inside of the cell, we're going to make it more positive than our negative 70, all right? Or you can say we're going to make it, all right, less negative. Okay, so that basically means we're going to go from negative 70 to negative 60, negative 50, negative 20. Okay, that's what we're going to do. Okay, how do we do that? We are going to open up those gated channels that we were talking about. Okay, chemically gated or voltage gated, doesn't matter. All right, we're going to open them up. Okay, there's a stimulus that we need to open them up. Okay, and we call that the threshold. More on that in a second. Okay? So, when we open up, all right, one of these channels, these gated channels, all right, for example, we're going to open up a voltage-gated sodium channel, it opens up. Where does sodium want to go? If it has the opportunity to move across the plasma membrane, where does it want to go? Inside the cell. So it's going to move into the neuron. It's going to bring its positive charge with it. So if it's moving into the neuron and it's bringing its positive charge with it, is it making the inside of the neuron more positive? Yeah. Yeah. Is that not the definition of depolarization? Okay, good. All right. So what will happen is when we open up these voltage-gated sodium channels, as it floods into the cell, your resting membrane potential goes negative 60, negative 50, negative 30, negative 20, negative 10. Then it becomes positive 10, positive 20, until it reaches this value here. Positive 30 millivolts. All right. At that point, if we're dealing with the sodium, the voltage-gated sodium channels, they'll close down. All right. So let me show you a picture. So for depolarization, okay, all we're going to do is we're going to drop our value from -70 to -60. And you can see when we opened up this gated this gated sodium channel, all right, sodium enters into the cell, and it made the inside of the cell more positive. Are we good with that? Okay, depolarization. Okay, all we do, even if it goes into the cell and it makes the inside of the cell -69, it's still more positive. -68, it's still more positive. It's still depolarization. Okay? All right. So now, all right, we have a cell whose membrane potential is -60. All right? I want to get it back to that -70. The term that we use is called repolarization. This is where we're going to return, all right, our membrane potential back to the resting membrane potential of -70. So what happens here? Okay? Well, we activate our voltage-gated potassium channels. They're normally closed, okay, until some sort of stimulus opens them up. And in this case, we know what the stimulus is going to be. Some sort of change, all right, in the electrical charge across the membrane. So we just went from -70 to -60. Would you call that a change in the charge? Yeah, you're darn right. Okay? So they're open, all right, and then what happens is, all right, potassium starts to leave the cell. It exits, stage right, okay? And as it leaves, it takes its positive charge with it, okay? So if it's leaving the inside of the cell, it takes its positive charge with it, all right? Is it making the inside of the cell more negative? Yeah, you're darn right it is. It's making the inside of the cell more negative. And we're trying to get back to this value here. So we're at -60, all right? In this example here, potassium channels open up, and now potassium leaves the cell. So now it goes from -60 to -62, then to -64, -68, then -70, all right? It's returning back to the resting membrane potential, okay? And then when we get back to that resting membrane potential, that causes these voltage-gated sodium channels to revert back to the resting state, which means the activation gate closes and the inactivation gate opens up. We'll get into the specifics. Trust me, I'm going to get real, we'll do a step-by-step story. Okay? So here's repolarization. All right, what we're seeing here, all right, this represents, all right, the voltage-gated sodium channels here. All right. Regardless, we went, all right, from our resting membrane potential of -70, it went to -60. All right, in that example, then it went down to, it shot all the way to +30. Okay. What that does is that triggers the opening of our potassium channel. And then our potassium then leaves the inside of the cell, taking its positive charge with it, and it starts to make the inside of the cell more negative, dropping our value back down to -70. That's repolarization. Okay, so notice with potassium channels, they only have two stages: open, closed. That's it. Okay, voltage-gated sodium channels have the resting, the activation, then the inactivation. So that's 3 stages, okay? But potassium only has the 2. It's either open or closed. That's simple. Keep it simple. All right. Then this last term here is called hyperpolarization. Now, hyperpolarization is when we make the inside of the cell more negative than the resting membrane potential. So that means it could be -72, -74, and the example here that we use is -80, okay? So we go from a -70 millivolts, which is our resting membrane potential, to -80. Now, what causes this, all right, is when we see that the potassium channels are open, all right, and we can also see an opening of, we haven't talked about the chloride channels, but they can open up. Okay, chloride channels are going to be found all right, over by the receptive segment of our neuron. Where's that? The dendrites in the cell body, okay? And so positive ion, all right, potassium leaves, but guess what? Chloride, look up on the board, there's more chloride, all right, outside the cell than inside the cell. So chloride wants to move into the cell. It's going to bring its negative charge with it. Potassium's leaving the cell. It's taking its positive charge with it. So the exchange of these ions, all right, are going to make the inside of the cell more negative, making our resting membrane potential, in our example here, negative 80. All right. Once these gates close, all right, then the sodium-potassium ion pumps will eventually restore our resting membrane potential because they're going to pump sodium out of the cell, okay, and it'll pump potassium back into the cell. All right, so that's this example here when we're talking about hyperpolarization. Two phenomena can occur, okay? All right, so we have our potassium leaving, and at the same time as potassium's leaving, chloride can enter in, and it's negatively charged. So that's going to make that membrane potential, all right, to negative 80. So how does depolarization and hyperpolarization occur in a neuron? All right. So when depolarization occurs, we're going to have our cation, okay? That can include calcium, all right, sodium, all right? Or when we get our voltage-gated sodium channels to open, all right, the positively charged sodium moves into the neuron, making the inside of the cell more positive. That's the definition of depolarization. We make the inside of the cell more, or the inside of the neuron more positive. Hyperpolarization, all right, we've got our potassium channels open, and potassium leaves, taking its positive charge with it, all right, or we can have our chloride channels open, all right, bringing its negative charge into the cell. What time is it? All right, I'm going to start talking about graded potentials only because we're building up to the big event. And the big event is the action potential, the nerve signal. All right, I had to teach you all this stuff first, all right, so you can understand, all right, how we set the table, because we're setting the table right now. It's not time to eat yet, but we're setting the table. All right, so now we're going to serve our appetizer. Okay, the main course is the action potential, the entree. Okay, but we need to start, all right, with something small. So we're going to talk about the graded potentials, right? And I'm not going to call them mini action potentials because they're not. All right. So right now we're going to start from for the rest of this chapter. We're going to start here, talk about the receptive segment, and then we're going to move on to the initial segment, to the conductive segment, and then to the transmissive segment. So I'm going to start by talking about what happens here. Okay. All right. So starting off in the receptive segment, we're going to talk about this phenomena called the graded potential. Okay. This is going to be our appetizer. So a couple characteristics that you need to understand about graded potentials. One is that they don't live very long. They're short-lived and they're small. Okay? So they're small and short-lived changes in our resting membrane potential. So remember how I was telling you, you can go from a negative 70, which is the resting membrane potential, to a negative 68? Okay? That could be a graded potential that caused that. All right? It's a short-lived, small change. Okay? These only occur in the receptive segment, which means can only occur in the dendrites, in the cell body, or the soma. Okay? Nowhere else are you going to find these graded potentials. All right? And what happens is we will see the opening of chemically gated channels. So if they're chemically gated, all right, the stimulus that we need to open them up is going to be a neurotransmitter. Okay? So a neurotransmitter is going to be released on to our receptive segment of our neuron, and it's going to trigger the opening of these chemically gated ion channels to let in a specific type of ion, either to let it into the cell or let it leave the cell. That's what we're going to see, okay? So, and it's only going to be a small amount, all right, for a short period of time. Very short, okay? All right, so a couple of these characteristics here of these graded potentials. All right, yes, I said they're small, but we can affect how big. I mean, it won't be as big as an action potential, but we can make them bigger depending on the number of channels that we open up. If we only open up one channel, it's not going to be big. If we open up 10, we'll make it larger, okay? So the degree on how we're able to influence, all right, these graded potentials, and the degree and influence of how we're going to change our resting membrane potentials definitely depends on the number of gates that we open up. A larger stimulus is going to open up more gates, okay? So this is a binary type of scenario, meaning it can either do one of two things. It can either depolarize the receptive segment, which means it can make the inside of the receptive segment more positive, or it can hyperpolarize, meaning it can make the inside of the receptive segment more negative, okay? Another characteristic is these ions, when they enter into the cell, they're going to move parallel to the plasma membrane. So here's the plasma membrane. They enter in, all right, and then they just start to move along like that. Okay, they're going to move parallel to it. Okay? Now, unfortunately, when an ion moves into the cell's filled with goop, right? The cytoplasm, okay, is made up of three things. One of those things is the cytosol. The cytosol is made up of a viscous substance, all right? It's going to be like swimming in a pool of motor oil. So these ions have to try to push their way through this motor oil, all right? That thickness of that motor oil creates resistance, okay? And over time, it becomes weaker and eventually ceases. So, easy way to understand that is taking a rock, throw it into the water, okay? The ripples around where you threw the rock into the water are pretty big, right? But as they move further away, do they not get smaller and smaller and smaller? The reason why is the energy waves, all right, experience more resistance. All right, not more resistance, but resistance, and over time, that resistance makes the energy weaker and weaker and weaker. Think of it like that, okay? So when the ions enter in and they start to diffuse into the cell and they start to move along the plasma membrane, or parallel to the plasma membrane, all right, the further they get from where they entered into the plasma membrane, the weaker they become because they're experiencing resistance. It's like swimming. You're not going to swim as fast, you know, an hour into when you started swimming. You get tired. Think of it like that. The ions, as they're experiencing resistance, become tired and weaker because they're losing energy. Okay? So you might be wondering, well, who cares? Remember, our goal in our neuron is to create an action potential. Where does that action potential occur? Where do we create the action potential? I'll even throw the map up there for you. Where does it happen? The axon hillock, right here. Okay? That's the initial segment. All right? This is the receptive segment here. So we need to get, if we have another neuron that is creating a graded potential here, it's got to make its way all the way to here. All right? So this whole time, we're going to be generating, all right, graded potentials. And our goal is to get these graded potentials to make their way to the axon hillock. And they're going to come from all different areas there. Okay? And our goal is to have either enough graded potentials or graded potentials that are powerful enough to cause or create an action potential. Okay? And I'm going to explain to you how we do that. Okay? But in order to understand that, you need to know these characteristics here all right, when we're talking about, because that's ideally what we need to do is we need to change, all right, our resting membrane potential to a specific value. We call it the threshold value. I'm just going to call it the T value for right now. The threshold value, you need to know this, is negative 55 millivolts. If we can get our membrane potential to negative 55 millivolts, and if we can get, all right, that value to occur in the axon hillock, we're going to make an action potential. Okay? So we need to go from negative 70 millivolts to negative 55. So that's a difference of positive 15 millivolts. Okay? But more importantly, we have to get, all right, that negative 55 value to occur here. Doesn't matter if it occurs here. That's not the axon hillock. All right? We need it to occur here. All right? And our graded potentials are going to help us to get there. Okay? We just got to get those signals and enough of them, and they need to be strong enough to get to occur or travel to here. But keep in mind, the more they travel, the weaker they become. All right? Like that, those ripples, all right, from the rock. They become weaker, all right, the further they move away. Okay? So we need to get those graded potentials all the way to the axon hillock there. All right? All right. Let's take a break because this next stuff will blow your mind and you're not ready for it yet. It's not, I don't have enough time. You don't have enough time to get that done. So let's take a break. If you folks at home have any questions, ask me in lab. All right? I'll see you in about 10, 15 minutes. Okay?